Hi, hello everyone. Yesterday I have discussed about the direct planting and transplanting of rice and the advantages and disadvantages of transplantation of rice. In today's class I am going to discuss about the transplantation shock. While transplanting seedlings, the seedlings undergoes transplantation shock that will occur while transplanting from nursery to main field. Okay. It takes 5 to 7 days to recover from transplantation shock. Okay. Rice plants transplant at the age of 21 to 25 days after sowing. As you know, DAS means days after sowing. Okay. For SRI, that is system of rice intensification, rice seedlings are transplanted at 10 to 12 days after sowing. About SRI, I will discuss in detail in another session. Now I am just telling that the transplantation duration for rice crop in different methods. Okay, for DAPAG method it is 11 to 14 days after sowing. DAPAG method also I will discuss in detail in another session. Okay. Actually, the nursery area required for transplantation of rice for 1 hectare is 1000 meter square. That is, we have to cultivate 1000 meter square nursery bed for 1 hectare. Okay. For system of rice intensification, it is 100 meter square. Yes, as I said, I will discuss later what system of rice intensification. Okay. Fertilizer requirement for rice crop is 100, 60 and 40. Rice prefer nitrogen uptake in ammonical form. Okay. The best fertilizer for crop dressing is ammonium sulfate. Okay. We found azospirillum, that is the bacteria found on root surface of rice for nitrogen fixation. Okay. Critical stage, that is plant require water in that particular stage, definitely, that is called critical stage. We should recommend irrigation in that particular period, that is critical stage. Okay, that is important stage to apply water. Or the stages in rice are tillering to flowering stage. Okay, those are the critical stages for rice crop. Okay. Generally we will go lowland rice in our areas. The upland rice will cultivate in mountain or hilly regions. In lowland rice, the fertilizer supplied in reduced zone. Reduced zone means the oxygen levels are very low in reduced zone, that is underneath the soil. Anaerobic layer, that is oxygen levels are very low, that is reduced zone. Super rice variety in rice is Lunishri. Okay. First intervarietal cross variety of rice is Jaya, that is the cross between TN1 and TN141. Gene responsible for dwarfness in rice is DGU gene. This is the most important bit. Most of the students know that this is a very most important bit. Tillage implement suitable for rice cultivation is power tiller. Okay. Hulling percentage for rice is 70 to 75%. In rice, among them, the most dominant weed species in rice is Echinoclover species. Khaira disease in rice is caused by zinc deficiency, and Kyoshi disease is caused by H2S toxicity. Okay, this is by zinc deficiency and this is by H2S toxicity. White ear of rice is caused by iron deficiency. Okay, in rice will grow for parboiling. Parboiling is the process, hydrothermal process of rice, which saves vitamin B12. Okay. Aroma in rice is due to the chemical diacetyl 1-propylene. Okay. Okay guys, that's all for today's video.